Welcome back to the Flagship Studio here at the JP Morgan Health Conference 2024. I am delighted to be joined in the studio by Mahesh Karande, the CEO of Omega Therapeutics. Omega made some very big news earlier this week with a partnership on obesity, the sort of area of incredibly hot focus in the biotech and pharma markets, with Novo, an incredibly hot company in that space. Tell us what Omega is going to bring to Novo and how the partnership will work.
Yeah, we are super excited about that partnership, Christine, and thank you for having me. This partnership is hugely validating for from Novo Nordisk standpoint for Omega, right? Omega's platform tackles some very fundamental biology. So if you think about what's happening in the obesity space, right? People are using GLP-1s, and GLP-1s are used sort of in a way where they're not treating the underlying disease necessarily, but treating symptoms. So people feel less hungry.
Right.
I mean, less hungry.
You're right, less hungry,
Right,
yeah.
exactly. Less hungry leads to satiation and also gastric emptying, right? But if you stop taking GLP-1s, right, the weight comes back with a vengeance. Exactly, with a vengeance. So if you think about a next generational drug, one of the areas of science which is super interesting is this process of thermogenesis. Simply put, it's turning white fat into brown fat. So babies are born with 100% brown fat, right? And as we grow older, we develop a lot of white fat, which essentially just sits around and leads to obesity, right? If there was a way to convert that white fat into brown fat, which then burns itself off, that's an irreversible process. This science is well known. It is very, very difficult to tackle this science with small molecules or potentially siRNAs or non-coding RNAs, right? So as you think about the landscape of types of drugs that you could develop, we come into the picture with a pioneering, approach doing epigenomic control, right? Where we are going fundamentally to where genes sit and we tune them to engender a biological process.
Epigenomic controllers is a whole new class of medicine. So say a little bit more, just go a little deeper on the fundamentals of the biology of how that works.
Yeah, so the epigenomic control is, if you think about how genes get expressed, as we are sitting here, as everybody's sitting around, right? Our genes are expressing and life is going on because, the central dogma of biology works. Transcription happens, translation happens, protein gets created. That's that's the building block of life, right? When disease occurs, those genes are either overexpressed or underexpressed. Or sometimes, in the case, for example, in thermogenesis, you need to recharge those genes to convert that white fat into brown fat. What we are able to, that's really what nature does. How nature works is nature lays these epigenetic marks at regulatory elements close to these genes that control gene expression, very simply like you would tune a thermostat, right? We figured that out and we deployed our technology, which is essentially mRNA technology. What we are doing is we are the first systematic use of mRNA therapeutics as programmable epigenomic medicines. So we are sending mRNA through the ribosomal machinery into the genome where it expresses two proteins. One is a DNA binding domain that goes into and homes into that regulatory sequence, which is unique. That's a huge target list that we have. That's proprietary to Omega, and there it lays an epigenetic mark that we decide based on the disease that we are treating, whether it's up or down regulation of the gene, and the duration of it.
So either the jumper cables or turning down the thermostat.
Exactly, and also the duration, because think about it, right? Diseases need to be treated differently. So in oncology, for example, where physicians treat patients once every 2 weeks, we have designed a epigenomic controller that lasts for 2 weeks, right? Now in chronic conditions, we have epigenomic controllers that we have already demonstrated with a single application working for 6, 7, 8 months. So that's really the powerful technology that we have, and we can develop these very, very rapidly. So our first IND took us only 27 months from scratch to getting it cleared. So this is the era of programmable medicines, and epigenomic control is really at the forefront of mRNA therapeutics being used as epigenomic medicines.
You mentioned clinical readout and clinical data. Not only were you fast in the clinic, you've got data. Tell us what you're finding and how it's giving you a sense of what comes next for the company.
Yeah, look, the clinical data is fascinating because, first and foremost, we put out data in September because what we saw is in the first 2 dose levels, 8 patients, the drug behaved exactly like it was supposed to behave, right? You don't usually see that in the clinic. We were blown away.
Sort of
right?
like Moderna's first trial with mRNA,
Yeah,
8 patients. Yeah.
exactly, right? And we are happy to follow on in Moderna's footsteps. But in all 8 patients, we saw the mechanism working exactly the way we wanted it, right? The drug being uptaken, the pharmacokinetics exactly like we expected, the pharmacodynamics as seen by the methylation, basically the epigenomic mechanism that we were tracking. We saw it exactly working in all 8 patients, and then we saw mRNA downregulation. So if you think about the central dogma, what you're doing is you are you are going and laying an epigenetic mark at the gene, in this case the MYC gene. You downregulated it to the level that we exactly wanted. We saw that in the epigenetic marks that were made, that we measured, and we saw that in the mRNA downregulation. That should lead you, obviously, to tumor killing. Now, as the trial progresses and as we get healthier patients in the trial, right? I mean, this is just the first phase. These patients don't have enough of a life expectancy that they can stay on the drug long enough, right? So
when will you broaden to a broader?
Yeah, so later
Yep.
on this, like as we have publicly said, we now have completed dose level 3. We are starting dose level 4, and then we'll continue to dose escalate, right? And at some point we will expand when we hit the right optimal dose.
Gotcha.
We are very optimistic based on the data we have seen, and then we'll also do a combination. So the beauty of this trial, by the way, is actually in the combination study where we have demonstrated in preclinical work that if you treat and this was in mice, but we treated 20 competent mice. The immune systems were perfectly working that were implanted with HCC tumors.
Okay.
We treated them with our drug, for a certain period, and we saw 70% complete response, monotherapy. And then what we did was we let those mice stay without drug for another period, right? And then we implanted on their flanks the HCC line that they'd seen, and on the other flank a non-small cell line that they're not seen. And we didn't give them any more drug. 100 days after their dosing was stopped, what we saw was the HCC tumors did not grow, whereas the NSCLC tumors grew. So this is exactly how the drug is supposed to work. Obviously, in monotherapy, we expect, but in combination, you are really, what you're doing is combination therapy will work as well, but what you're doing is you're changing the tumor microenvironment, where we hope to demonstrate in the clinic that after a certain period of time, the host immune system should start taking over and actually killing tumors. So this is the promise of epigenomic control. This is the promise of Omega Therapeutics.
Exciting new partnerships, encouraging data. 2024 promises to be a big year for Omega Therapeutics. Thank you for joining us in the studio.
Thank you, Christine.